A-level biology, going through topic by topic. Today we're going to have a look at the specific immune response. We're going to start looking at these things called antibodies, which do all the hard work, and how they're basically controlled, looking at the specific immune response. Let's start with having a look at what an antibody is and what it does. So let's see what we can do with this. Right, now let's start with drawing what one of these antibodies might look like. And this antibody consists of basically a sort of a Y chain. And this is sort of mirrored here. Then we've got something which is called the light chain, and this goes here. So let's have our light chain, and I suppose let's make these a bit clearer. These chains are held together by disulfide bridges, and we've got one of these disulfide bridges here. And they hold this like that. Now, all of these antibodies basically look like this. So this bit here, this is called the constant region. And all the antibodies basically look like this. Now, attached to the end are some bits which now vary, and these are basically specific to the particular antibody that we're trying to sort of make. And so let's draw this one in. Now, because this bit here is our variable region, let's push it across a bit. This is these bits here. This makes up what all the different antibodies basically look like. And what we get up here, this is our binding region. So this is what actually binds to a particular antigen. So I've got a particular antigen, and this is specific to that, and so this will hold on to the antigen. So the way that we can think of that these work, these bind with, if you like, a lock and key type of system. So our antibody here that the body has made is very specific to a particular antigen. Now, basically here once we've managed to get this antigen antibody complex, this acts to attract the phagocytes, which come in, and they are then engulfed by the phagocyte macrophages, and they are basically digested, destroyed. Another way of looking at this is this antigen, perhaps this protein on the surface of a virus, now can't cope terribly well if we've got one of these things attached to it. It can be worse though. Let's look at something like a little virus, and let's imagine this virus has a specific here, we'll call this its antigen. We've got an antibody that will respond to this, and this is going to sort of bind here to this, so that's going to bind to that, and we've got our specific here Y shape here, and you'll see the reason for all the effort of trying to sort of do this in a sec. So I've got this, then we've got again this particular region here. So this is just here one antibody, but now it can collect up more than one virus particle, and worse for this particular thing here, it may give us another one that we can sort of stick on here. Like this. And I can get whole clumps of these. So what this is going to do is we're going to act as, the proper name is an agglutinin. So this is going to basically cause clumping together. And of course, if this is happening, we're managing to take out whole loads of these different virus particles at the same time, because one of these is going to bind to probably one, but maybe two of these different antigens on a virus surface. In the same way, if we've got a toxin, some sort of little chemical that's causing some problems, so perhaps this is our chemical that's causing problems, then if we've got this sort of antibody here, or maybe several of these sort of connected to our particular chemical, I can sort these out. Yeah, like this. Oops, drew it the wrong way. Then basically our chemical could be taken out of action because basically it can't do anything if we've got a huge number of these chemicals sort of attached to it. So my chemical here is literally engulfed, and this toxin that we've got here is effectively neutralized. So this antigen here is acting as an antitoxin because this chemical now can't combine with anything else because it's got these huge things attached to it. And this sort of does cause problems for the bacteria because their toxins are being stopped and these antibodies are connecting to the surface of bacteria or viruses and basically clumping them together and stopping them work. So we've got, if we look at this molecule, we've got some bits and pieces that we tend to name. These here are called the heavy chains, and these don't change. We've got this region here which is called the light chain, and again these are part of the constant region that all antibodies contain. This, they look like this. And then we've got this region up the top which is very specific, and this one that's specific is our variable region. And that can make all sorts of different types of surface here, and I believe that the fact that we've actually got two chains enables basically a wider range of shapes, and typically in a human you're going to have at least 1 million different ones of those in your body at any time, at least that number. So these antibodies, and I've got a sort of an idea of what one might sort of look like, and there is sort of an idea of a three-dimensional shape to try and give you an idea of what these things look like. So these things literally go around the body in their millions, going round and literally sticking to anything. Think of them like sort of glue or flypaper, and as they go around they're just literally sweeping up those molecules, those viruses, those bacteria. And the bacteria find it really hard to sort of do much if you've got this enormous thing attached to you. And by having more than one of these like this, we're getting this agglutination. which is basically stopping these things sort of moving around and doing anything. Now, how can we do some of these things like antibody tests? Well, let's have a quick look and see how one of these antibody tests might work. The antibody test can be done perhaps in one of two ways. So let's think of having perhaps our surface, and our surface has got probably these receptors on them, the things that we're looking for. We'll call this, if you like, our antigen target, and our antigen attracts our specific antibody, which is now going to bind to this. Another one out back. I'm trying to be consistent in the way I sort of draw these, and what I could perhaps do is get something to bind here, and if I can get something to bind here, this could be some sort of radioactive, fluorescent, some sort of dye, some sort of means of identifying. So we can actually then do some sort of identification to show that this has stuck on. Doesn't have to be done like that. That is what we call doing a direct immunoassay. Because basically this antibody binding to this antigen, and then I can stick some sort of, basically, I'm going to call it a name, I'm going to call it a reporter molecule. And as I said, this reporter molecule could be something like radioactive, fluorescent, coloured, anything, so we can try and identify what it is. Alternatively, we can try this a different way, and if I've got another one of these molecules coming along, I think I've drawn that around the wrong way, because I doesn't matter. Got my molecule. And then what I could do is I could produce a different antibody, trying to show you it's here different by simply putting on different colours here to try and give you an idea that these are slightly different. And I could bind, if you like, this one, and we'll call this one antibody A, which is going to seek out the specific antigen target. And then I could have basically here some sort of secondary antibody, and this would be an antibody that would bind specifically to the antibody, and this would stick on, and this has got again some sort of reporter molecule on it, and this would be more of an indirect immunoassay. So I can either bind directly, or I could then add a second antibody, and maybe this one we have stuck to something, and that would be like perhaps maybe a pregnancy test. But we can use this in any form to try and identify a particular virus, be it COVID-19 or be something like a bacteria like tuberculosis. It could be anything like this to try and identify what's going on. So there's how the antibodies basically try to defend the body, and we've got these systems used for all sorts of various bits and pieces. So we can use these made as what we would call monoclonal antibodies, so growing up hundreds, thousands, millions of this one specific antibody, and we can use this for things like medical diagnosis. So we can use this to look for things like influenza, obviously the one that's working at the moment, the coronavirus, but it could also be other things like hepatitis, and other ones like, I suppose, chlamydia, various things to try and do this, but as well as those things, we could perhaps get it to look for something like PSA. PSA is the prostate specific antigen, and this will be possibly in high levels in someone that may have prostate cancer. So if they've got, prostate problems, then that could be noticed. It's also going to work well in pregnancy testing. Simply you're looking for a particular antibody or an antigen. It depends what we want to just term what we're looking for. In this case I've made an antibody, but it is an antigen to that antibody. Now, are there any ethical uses of some of these things? Yeah, there are, and because of that it means that we can sort of raise some important issues here. So what would be our ethical issues? Well, many of these are made with monoclonal antibodies, and many of these are made in mice, and these mice are used to produce both the antibodies and also producing tumour cells in mice. And you know, is this going to kill the mouse? Yes. Is it going to damage the mouse? Yes. And they're collecting the antibodies, the monoclonal antibodies. Is this going to kill the mouse? Yes. And is this right? We've got it killing, destroying various nasty sort of diseases, but we do have to worry about, can these monoclonal antibodies go off and do anything else? And the answer is to this, well, maybe. Testing drugs also has a problem with rather different sort of safety things, and maybe if we're not careful we can cause problems again with this, that it might start to sort of raise issues about how and where these things are used. This isn't biology, of course, this is sort of going in, having a look at ethical ideas. So there's some things about antibodies. Now, antibodies are actually made in specific cells in the body, and what I want to do now is have a quick look at some of these, the T cells, the B cells, basically the white blood cells in the body. So let's have a look and see what we can do about some of these and get some ideas about what's going on. So let's have our specific little virus down here, and this virus has got on it some of these little diamond shaped little bits and pieces, and this is going to be engulfed by one of our cells, and this cell is going to basically engulf this, and this cell is going to be one of our phagocytes. Going to take this in by phagocytosis. So now I've got this little critter inside, and we're going to bind this to a lysosome, and that's going to get in and destroy that. But what will happen is this particular antigen can be presented on the surface of this phagocyte. Along comes a particular cell, and we're going to call this a T helper cell, and this T helper cell is going to try and basically find something that is going to bind to this particular antigen, some sort of antibody. And this cell will then basically be, when it's found something, cloned by mitosis. And what we've got then are our nice little T cell. This T cell can go off and develop into a memory cell. As a memory cell, it's going to remember that particular antibody for that particular antigen, and what it's going to be able to do is, if we get some sort of infection in the future, we're going to sort of get things working fairly quickly. So we would call all of this cell mediated immunity. Now, as well as producing the memory cell here, we're going to produce chemicals that will stimulate basically the phagocytosis, and we are going to produce special T killer cells. So we've got quite a few different bits and pieces here, and we're also going to here stimulate some B cells to divide. So I suppose I better mention now what are the difference between T cells and B cells. So we've got B cells. These are made surprisingly in the bone, and they're made in the bone marrow, and these particular cells are our white blood cells, and they go round and they do all sorts of damage to particular bacteria and viruses. We've got T cells as well. Now these are made in the bone, but then are, if we like to think about, they're finished off in the thymus gland. When I talk to many students, they've got absolutely no idea where their thymus gland is. Just here. And it's often seen when you do something like a lung dissection. You often get a thymus attached to that. So we've got these two cells, B cells made in the bone and T cells elsewhere, the thymus. So that's cell mediated immunity. Now, cell mediated immunity, basically here, T lymphocytes respond to cells in the organism that have been changed in some way by some sort of virus infection, by an antigen, by a mutagen or something. And cell mediated response is particularly important against viruses and in fact some early cancers. And this is some of the non-specific defense system. These macrophages, these phagocytes here, engulf and digest the pathogens and present them on the surface. These have got a special name, these cells, and they often get called APCs. Antigen presenting cells. And then the receptors on some of the T helper cells here, they fit the antigens. These T helper cells become activated and produce some chemicals called interleukins, which stimulate more T cells to divide rapidly by mitosis. They form lots more identical cells here, and they all carry the right antigen to that particular pathogen, and then these clone T cells can go through and make either memory cells, which give rapid response to this pathogen if it comes again. They produce this phagocytosis, they produce more B cells dividing, and they develop into these T killer cells. So let's now go and have a look at a different type of immunity, and this is called humoral immunity. Here we are going to have our particular evildoer, and this evildoer has got with it some antigens here, and maybe a few of these have broken off. Along comes a B cell. Whoops. So along comes here a B cell, and it's going to engulf and necessarily destroy these antigens, and again it's going to present this antigen on the surface. So this cell is an APC, an antigen presenting cell. So we've got these on the surface, and not just one but a whole load of these. Now what's going to happen? Well, at this point we think of a humoral activity, immunity here, this activity here. The body responds to antigens found outside the cells. These are produced by things like bacteria, fungi, and that sort of stuff. The humoral immune system here produces antibodies that are soluble in the blood and the tissue fluid and are not attached to cells. The B lymphocytes here have antibodies on their cell surface membranes, and there are millions of different types of these B lymphocytes, each with a different antibody. And when an antigen on the pathogen enters the body, the B cell with the complementary antibody binds to it. It engulfs it, and in the process the antigens here are presented on the surface. Now along comes one of these activated T helper cells, and this activated T helper cell here basically has got the right sort of clonal selection at the point at which this B cell has got the correct antibody to overcome a particular antigen. So interleukins are produced by this here, which basically are doing jobs like activating these B cells. Simply by having this sort of binding here. So off we go with our cloning. So we've got our cloning by mitosis, and this is going to produce another cell here, and this one is producing loads of these antibodies, which are then released into the cytoplasm. So this is just a plasma cell. And of course we've got a memory cell. So these cloned plasma cells produce antibodies that fit the antigens on the surface. They bind to them, they disable them, as we've looked at, and basically these can take, I suppose, generally we think of these taking days or worse, perhaps weeks to get going, become fully effective against that particular pathogen. And that's why you get ill. The symptoms are the result of the way our body reacts when these pathogens are dividing freely before our primary immune response becomes fully operational. And if it's taking many days or even weeks to get going, if this pathogen is effective, then it can kill you before you've managed to create enough of these, which is why in this battle against COVID-19, the coronavirus, now we're trying to extract some of these antigens from people who've already suffered and try and put them into the body of those that are ill. If, however, you get this thing again, then, and our hope is this is going to work with this coronavirus, then you can produce from these memory cells this number of antigens very, very quickly before basically you can get even the symptoms of the disease. That's the theory. But there can be one little problem with this, and that is something which we're going to call an autoimmune disease. In an autoimmune disease, basically the immune system stops recognizing self. So we have a little flag on a cell, and this flag here says, this is me. And somehow what happens is that you can get either antibodies created or just killer T cells that will just literally produce these antibodies that bind to self. Now, exactly why this happens can be either due to perhaps some sort of virus, and HIV is one of those that we know that does this, but exactly how, that's a different problem. There are basically, as far as we know, somewhere around about 80 different autoimmune diseases, which we call AIDS. And basically the only way to try and survive these are to use immunosuppressant drugs. In other words, drugs that prevent this immune system working. And that works. The only problem, of course, is that this now means that you're not going to survive things like all this COVID-19 or whatever you're going to get. So there are certain types of immune diseases. Let's just mention 3 of these. So we can get type 1 diabetes, and in type 1 diabetes it's our insulin secreting cells, and these are being targeted by this autoimmune system. So your immune system is actually attacking your insulin secreting cells in the pancreas. How do you get around this? Well, we can have, if we want, some sort of treatment. We've got insulin injections just to replace what we've lost. You could try a pancreas transplant, and it may work. Or we can try these immunosuppressant drugs. Another one of these is rheumatoid arthritis. Basically, this is going to affect all your joints, the hands, the wrists, the ankles, the feet, and unfortunately for this one, there is no cure. All we can try and do is try some immunosuppressant drugs. They might work. Steroids are known to sort of help a little bit, and anti-inflammatory drugs, but we are going after basically the symptoms. We aren't finding a cure for this. And another one would be lupus. Here we've got affecting skin, joints, fatigue, and any particular organ in the body can be affected: the kidneys, the liver, the lungs, the brain. And again, here we're looking at no cure for this. Anti-inflammatory drugs, steroids, immunosuppressants, can try all sorts of things, but these are our autoimmune diseases where we are basically attacking ourselves. So there we are, some ideas on antibodies. Also, we've got some ideas about cell-mediated and humoral sort of defense systems and the problem with autoimmune diseases. Thanks for watching. I hope you've enjoyed A-level biology, doing it topic by topic, and I will see you again next time when we'll have a look at whether we can actually perhaps prevent or treat some of these diseases. Until then, bye-bye.